Yeah, I have I could go for an hour, and the rest I could probably do with this. Yeah, it's a smartwatch. This one too. Smile. You're on camera. Yeah. I can show you something cooler. See this? It costs $5. Okay, I kind of like wrecked it, but it costs $5. It films here, so you just press this button, it turns it off, and this is your USB charger. Like less than $5 from China.
Changed the lecture notes a little bit. So there's a new set of notes. They're slightly different and there's some changes to last week. So go on the old one.
So, for those who were not here, the results of the exam will be next week. We get all the marks. So, today I'm very happy to welcome Elliot Brodetsky. He's associate professor at full professor, sorry. Professor
I forgive you.
at Hôpital Maisonneuve-Rosemont. His main research interest is to understand the molecular mechanism of nucleotide excision repair, and therefore he is very well positioned to give you a lecture on DNA repair. So thank you very much for giving this lecture. And,
I look so young, it's natural to think I'm an associate.
sorry,
It's okay.
Okay, enjoy.
Okay.
Bye.
Bye. Okay, now that that's over with, this is what we're going to be talking about today. I'm going to try to give you an overview of DNA repair mechanisms, but as I go along, I'm going to try to stress certain important concepts, and that will lead into a real-life example of why DNA repair is important in the context of cancer. So this is who I am. Whoops. Okay, Elliot Brodetsky. I did my postdoc in France. They called me Gros Biscuit. So, but you can't call me that. And this is where I am, and you can. This thing doesn't work very well. I'll do it with two hands. Yeah, and you can get in touch with me anytime. If there's something you don't understand, or if you have something intelligent to say that you do understand, I'd like to hear it too. See if this works better. Yeah, this works better. I might get confused though. Anyways, so, okay, we might as well start. Okay, I think a lot of you already know this, that DNA damage and genomic instability in general drive the process of multi-stage carcinogenesis. And that starts with a process called tumor initiation, where you have DNA damage, can either be from endogenous sources within the cell or exogenous environmental mutagens causing mutations in any one of many oncogenes or tumor suppressor genes. Then once you have the mutations in these critical genes, they usually or often encode proteins that are involved in apoptosis, cell cycle arrest, and DNA repair. So you have mutations in these processes, and this drives up the level of genomic instability in the cell. And upon more DNA damage, you get increased genomic instability and mutations. And you get tumor promotion. Now, a very good example of tumor promotion after DNA damage would be you have apoptosis deficient cells that have been initiated at the tumor initiation stage, and these will undergo clonal expansion when severely damaged at the expense of normal cells, which will die. So you have this clonal expansion of apoptosis deficient cells getting further along the road to cancer. And then upon even more DNA damage, more genomic instability, more mutagenesis, in this tumor-promoted population, you get the establishment of genetic variants that have increasing metastatic potential. This is what we call tumor progression. And then once you have a full-blown cancer, a full-blown metastatic cancer, it's kind of ironic that often DNA damaging agents are used to treat. The same DNA damaging agents that drive cancer are often used to treat it. And they can be very effective. They can result in prolongation of life or even sometimes a cure. Of course, there's problems associated with chemotherapy, such as clinical resistance and the toxic side effects of many of these DNA damaging agents. They affect normal cells as well as cancer cells, and that will be a bit of the theme that we're going to talk about. And if this wasn't bad enough, the clinical resistance and the toxic side effects, most DNA damaging agents, chemotherapeutic drugs that are used for tumor treatment, because they damage DNA, they also induce mutations and cause genomic instability, and you can get the induction of secondary mutations, which lead to a new round of tumor initiation. So you can get the induction of secondary tumors that have nothing to do with the original primary tumor that are actually induced by the treatment. So this is kind of a vicious cycle of DNA damage and multi-stage carcinogenesis. So fortunately, there's a DNA damage response that protects cells against genomic instability and cancer. And we're not going to talk too much about this DNA damage response. Basically, when cells are challenged with DNA damage, there are proteins or protein complexes that will sense the damage. Following this mechanism along, you have mediators or transducers that are kind of preeminent, usually kinases, that undergo signal amplification and transmission, and they will often phosphorylate effectors, hundreds of effectors that regulate many different processes. And we're going to be focusing on DNA repair, but often these processes are not completely independent. There's some interaction between them, obviously, but again, we're going to focus on as much as possible on DNA repair, but we will be referring also to replication fork stability, and transcriptional activation will actually play a big role in the talk and what I'm going to tell you today. So I guess remember, DNA repair and replication fork stability are the two important themes that we're going to explore. So the DNA damage response is highly variable depending on the type of DNA damage, and indeed specific DNA repair pathways exist that remove distinct types of DNA damage. And I'll just go over these quickly. We'll be talking about most of them today, not all of them, but most of them. DNA double-strand breaks is an extremely lethal form of DNA damage that's induced by ionizing radiation, which can come from environmental sources, but also from medical imaging, and is used, of course, in radiotherapy of cancer. Also, certain chemotherapeutic drugs, like camptothecin and bleomycin, use DNA double-strand breaks. And there's a pathway, which we'll talk about today, collectively called double-strand break repair, which comprises two pathways, homologous recombination and non-homologous end joining. Then there's another pathway. This is the one that I know the most about. It's the main reason why I'm going to talk about it today. That repairs helix distorting what we call bulky DNA adducts, and I'll explain in a moment what a bulky DNA adduct is. And these bulky DNA adducts are generally induced by numerous environmental mutagens and certain chemotherapeutic drugs, and they're repaired by a pathway called nucleotide excision repair, or NER. And also another pathway that deals with this type of damage is called translesion DNA synthesis, which I'm going to describe that too. It's not really a repair pathway. It's more of a pathway whereby cells tolerate DNA damage rather than remove it by bypassing it during DNA replication, but it's still important to know in the context of today's lecture. And then of particular importance is base excision repair. Now, BER, or base excision repair, repairs oxidized bases, which are induced principally by reactive oxygen species, many of which come from endogenous sources, such as oxidative respiration and inflammation, and also there's some exogenous mutagens that can induce reactive oxygen species that result in the oxidation of DNA bases. You can also have alkylated DNA bases, which arise in various ways, but of particular importance here are single-strand breaks, particularly unligatable ones. So if you have a single-strand break that has a 5' phosphate and a 3' hydroxyl, it's very easy to be rejoined by ligase, but single-strand breaks can be generated that have these weird termini. They can be induced also by reactive oxygen species, and there's a pathway which is a sub-pathway of base excision repair called single-strand break repair, which repairs single-strand breaks. Then there's a pathway, which I'll talk about briefly today, DNA interstrand crosslink repair, which repairs lesions that covalently link bases on the opposite strands of DNA, which is obviously something that's very lethal. And the pathway is called interstrand crosslink repair, and it's actually the concerted action of three pathways: homologous recombination, nucleotide excision repair, and translesion synthesis. And then finally there's this pathway, which I won't talk about today because there's enough pathways to talk about. It's called DNA mismatch repair, but it's still a very important pathway in the cell that corrects DNA polymerase errors. So during DNA polymerization, if a G mispairs with a T, mismatch repair will recognize that mismatch, excise it, and put in the correct base. So that's a very important pathway, which we won't be talking about that today. We'll be focusing mainly on double-strand break repair. I'll be talking about this because I know a lot about it, and single-strand break repair are important. So double-strand break repair and single-strand break repair are kind of the most important pathways to know at this point for this lecture. So, these three handsome gentlemen, I don't know if you follow the news, but the recent Nobel Prize in Chemistry was announced, and it was awarded finally, jointly, for mechanistic studies of DNA repair. And as you can see, like myself, most people in this field are very handsome, good-looking fellows. But these individuals really, as most Nobel Prize winners have done, and I'll refer a little bit to their work. When the appropriate pathway pops up, you'll see their handsome faces again. But these people have contributed tremendously to the DNA repair field, and in terms of publicizing its importance and elucidating the mechanisms of DNA repair. So, what is a bulky DNA adduct? So bulky, I think you know what bulky means in French. It's costaud, I think, costaud, bulky, like me. So bulky DNA adducts are induced by a wide variety of comprise a wide variety of structurally diverse DNA adducts that are induced by many, many environmental mutagens and carcinogens, and also certain chemotherapeutic drugs. So examples are ultraviolet light, which is the main mutagenic component of sunlight, which we all know induces skin cancer. Then there's another very, very ubiquitous mutagen, benzopyridine diallofoxide, which is present in all cooked meats, anything that's burnt essentially, especially cigarettes. When you burn organic matter, you get benzopyridine diallofoxide, and it's the principal carcinogen in cigarettes actually, and of course that leads to lung cancer. Aflatoxin B1 induces an adduct that causes its presence, it's a dietary contaminant. It's present in, it's found in peanuts or stored grains, mostly in third world countries when they're not stored properly for long periods of time at the wrong temperature. When it's too moist, a fungus will grow on the peanuts or grains, producing aflatoxin, and it's actually probably the main cause, aflatoxin poisoning along with hepatitis virus infection is probably the most important cause of liver cancer in countries where it's epidemic, like China, for example. Liver cancer is extremely frequent, and it's due in large part to aflatoxin contamination. And then there's a chemotherapeutic drug, cisplatin, which is a very important anti-cancer agent. It's particularly effective in treating testicular cancer and also ovarian cancer, although there are problems with clinical resistance, but it's an extremely effective but also extremely toxic drug. So if it's administered to patients, it can wipe out their cancer, but it can also sometimes kill them, unfortunately. So I'm going to be referring to the main, to ultraviolet light, which is what I've been studying for a long time. So ultraviolet light from either a lamp on the bench in the lab or from the sun will produce ultraviolet light will be absorbed directly by the sugar phosphate backbone, direct absorption of UV photons. And this type of lesion always occurs between adjacent pyrimidine bases, either two thymines, as we have here, or it could be two cytosines, or a cytosine and a thymidine, but it's always two pyrimidines. And following the direct absorption of ultraviolet light, you get two major photoproducts. This one is by far the most important, and I'll be talking about it when I refer to UV damage. I'll be talking about the cyclobutane pyrimidine dimer, or CPD. But there's also a 6-4 photoproduct, which is important. And both of these lesions are covalent linkages. In the case of the CPD, it's a 5-5-6-6 covalent bond between adjacent pyrimidine bases. And normally, of course, the pyrimidine bases are not joined by covalent bonds, adjacent bases. And because of this, this interferes with Watson-Crick base pairing, and the principal property is that it's helical distorting. So this bond between these two bases, rather than the bases being like this, they're kind of like this. So a major property of bulky DNA adducts is that they distort the DNA helix, and you'll see why that's important. They also block DNA replication, which is also an important theme in this talk. They block transcription, because RNA polymerase II cannot get by it. It represents pretty much an absolute block to RNA polymerase II progression. And of course, you can imagine if the helical distorting block DNA replication and block transcription, these lesions are going to be highly genotoxic and carcinogenic. And in fact, as I just mentioned, excessive exposure to ultraviolet light, leading to the induction mostly of cyclobutane pyrimidine dimers, leads to mutations in oncogene tumor suppressor genes, and it's the preeminent cause of skin cancer. So if you stay out of the sun, you won't get skin cancer. If you take a lot of sun and you have fair skin, then you're asking for trouble, it's just like smoking cigarettes for your lungs. UV light is extremely mutagenic, and you should stay out of the sun as much as possible, especially if you have fair skin. Especially because at 12 noon in Rio de Janeiro, you get 100,000 cyclobutane dimers per skin cell per hour. And that's a lot. So I didn't want to scare you too much. Okay, so I mentioned that they're replication blocking. cyclobutane pyrimidine dimers, but many different types of DNA adducts are replication blocking. So replication blocking adducts cause what we call DNA replication stress, because DNA polymerase will encounter one of these adducts and it will stall, and there are various processes by which replication will restart, allowing cell survival. So the cell is under stress, and of course this happens during S phase, when DNA replication occurs. And the main cellular response to DNA replication stress is the rapid activation of this kinase, ataxia-telangiectasia and RAD3-related kinase, or ATR. Perhaps many of you have heard about it. Here, by the way, when I have these, on many of the slides, these references, these are, in most cases, really excellent references where if you want to get more information about what's on the slide, you can go, read either all of it or parts of it, and it will clarify things that may not be clear. But for the exam purposes, you're mainly responsible for what's on the slides, more or less. I'm not going to ask you tons of mechanistic questions and expect you to know the names of a million proteins, that's for sure. So in any case, this ATR kinase, here you have a very poor representation of the replication fork. Sorry, I'm not good at drawing all of those fancy things. But this is here you have the leading strand here on the bottom and the lagging strand on the top. You can imagine that. And you have DNA polymerase eta, which is replicating the leading strand. It's the leading strand polymerase, and it encounters a cyclobutane dimer here, or any adduct that blocks DNA replication. But the model that I use is the cyclobutane dimer, and it stalls. It can't get past this CPD. So what happens is that there's a helicase here, the MCM helicase. I didn't picture it. So the current model states that although the polymerase is stalled, the MCM helicase will continue to unwind the DNA ahead of the stalled replication fork, and that causes large tracts of single-stranded DNA to be formed. And these large tracts of single-stranded DNA will become bound by a single-stranded binding protein complex called RPA. Perhaps you've heard of it. RPA is the classical single-strand binding protein. So these tracts of single-strand DNA become coated with RPA, and that acts as a platform for the recruitment of ATR kinase and also various other proteins. I won't get into the mechanism here, but various other proteins that potentiate the activation of the kinase activity of ATR. So you'll have activated ATR after the assembly of multiple proteins on the RPA platform. Activated ATR then either itself directly phosphorylates or in many cases through the intermediacy of the immediate downstream effector kinase CHEK1, so it will phosphorylate CHEK1, which has a very strong kinase activity. That in turn will also phosphorylate many proteins. So either directly or through the intermediacy of CHEK1, many of these protein substrates will participate in what we call the DNA replication checkpoint, which induces a cell cycle arrest in all phases of the cell cycle. Also inhibition of late origin firing during S phase, so it slows down DNA replication in S phase, of course. And the main function of the DNA replication checkpoint is to stabilize stalled replication forks and to facilitate the restart of replication. Because if replication doesn't restart in a timely manner, what you get is what we call replication fork collapse, and this leads to replication-associated double-strand breaks. And I'll be talking a lot about this concept of replication fork collapse and double-strand breaks that are caused by replication fork collapse. Now, I'll say right off the bat that most people use this term, replication fork collapse, but nobody really knows what it is, okay? So when you have a stalled replication fork, There's one, Stephen Elledge, who wrote he's a great scientist, who actually he was the first person to characterize ATR kinase, and he calls that S phase a time when cells are playing with knives, because when you have an advancing replication fork and it stalls, there's a lot of DNA modifying enzymes, DNA repair enzymes, topoisomerases, all kinds of enzymes that are cutting up the DNA, and if the replication fork is stalled and it's not advancing, then you have all of these cuts and it just sometimes just falls apart. And when it falls apart, that's what people call replication fork collapse. It's more complicated than that, but that's the idea, and then you get these double-strand breaks. So this DNA replication checkpoint is very important, but what ATR also does is it phosphorylates many proteins that are involved in DNA repair pathways, like nucleotide excision repair, translesion synthesis, homologous recombination, and these DNA repair pathways are obviously very important for restarting replication because they remove the damage, and if you remove the damage, then the telomerase could just keep going. So obviously that's a good thing to do also. So you have multiple mechanisms that are promoted by activated ATR. So mainly replication fork stabilization and DNA repair to prevent this replication fork collapse and the replication associated double-strand breaks. So nucleotide excision repair, which removes replication blocking CPDs. This is a great review. If you want to learn about nucleotide excision repair, just read this and you'll know everything about it. Nucleotide excision repair, so some of you have seen this, a few of you, because I teach other courses. So if you've seen this, it's only the first part of the lecture that you've seen, so you won't get too bored. Nucleotide excision repair is the only pathway in humans for removing really any DNA lesion that distorts the DNA helix. That's the principal property of the lesion in order to be recognized by nucleotide excision repair. It has to be helical distorting or block transcription, and we'll see what I mean by that in a moment, why those properties are important. So nucleotide excision repair is comprised of two highly conserved overlapping subpathways. The first is global genomic NER, or GGNER, and this repairs helical distorting DNA adducts anywhere, absolutely anywhere within the nuclear genome. The second subpathway is called transcription coupled nucleotide excision repair, or TCNER, and this repairs transcription blocking DNA adducts, which are also helical distorting, but they happen to be transcription blocking as well. And they repair these adducts only along the transcribed strand of active genes, which is only about maybe 2 to 5% of the genome at any given time. So transcription coupled NER is a very specialized subpathway of NER that only removes DNA adducts on the transcribed strands of active genes that are producing a messenger RNA. Another important thing is that transcription coupled repair removes DNA lesions much more rapidly than GGNER, and this is what we call the preferential repair of the transcribed strand of active genes, meaning preferentially over the rest of the genome. So the way nucleotide excision repair works, there's two subpathways, as I mentioned. They differ only in the manner of lesion recognition. Only in the matter of lesion recognition. So I'll go over it quite quickly. In the case of GGNER, the global genomic NER pathway, you have a CPD indicated here as a red blotch, which distorts the DNA helix. This is a very bad representation of a CPD in a distorted helix. I'm sorry, but that's what it's meant to show. So you have a cyclobutane dimer which distorts the DNA helix. That will stimulate the recruitment of two proteins called DNA damage binding protein 1 and DNA damage binding protein 2, DDB1 and DDB2. That in turn will be followed by the recruitment of a protein, the trimeric complex XBCHR23B and centrin 2. So this is how lesion recognition occurs anywhere in the genome by GGNER. The transcription-coupled NER pathway is triggered by blockage of RNA polymerase II on the transcribed strand of an active gene at a site of a cyclobutane dimer. So RNA polymerase II hits the cyclobutane dimer and it can't go anywhere. It's absolutely blocked. It cannot get by. So obviously that's a serious problem in terms of maintaining transcription. After these two, the blockage of transcription by RNA polymerase II then results in the recruitment of two key proteins, Cockayne syndrome A and Cockayne syndrome B. So this is the lesion recognition step, either of GGNER and TCNER, and you can see they're different. In one case, it's the helical distortion that acts as a signal. In the other case, it's blockage of RNA polymerase II on the transcribed strand. Different proteins are recruited. However, the rest of the process, whether it be GGNER or TCNER, is identical. It's called the core NER pathway. I hope it didn't get cut off there. It looks like it did a bit. The core NER pathway consists of sequential steps where the DNA is unwound in the vicinity of the lesion, and this is catalyzed by the two components of the TF2H transcription factor. This is the same TF2H that regulates basal transcription, but it has a completely distinct role in nucleotide excision repair. It contains two helicases called XPB and XPD, and these helicases will unwind the DNA and open it up and make a bubble structure. And this allows the recruitment of other proteins that verify the lesion. Here you have RPA again. RPA is the single-stranded binding protein which will bind to the undamaged strand. It's now in a single-stranded state. And XPA protein will bind to the lesion. So we call this lesion verification because this is believed to mark where the lesion is. It's on this strand and not on this strand. So it kind of positions the next step, the proteins involved in the next step, which do DNA incision. And these are XPF ERCC1 and XPG, which are structure-specific endonucleases. They're recruited. They make a cut on either side of the cyclobutane dimer on the same strand. It's called damage incision. And this results in this cut permits a small nucleotide. It's about 30 base pairs apart, these cuts. So a small oligonucleotide containing the lesion is released, probably just floats away. And that leaves a gap in the DNA. So that gap is filled in by normal DNA replication factors. We call it DNA resynthesis. It's about a 30 base pair gap again, and it's filled in using the undamaged complementary strand as template. And then finally, the nick, the open nick here, will be sealed by one of two ligases, DNA ligase 1 or DNA ligase 3. And this is a completely error-free pathway. Once this process is finished, the integrity of the DNA is restored in a completely error-free manner. It's a perfect process. That's why I chose to study it, because it's perfect. And it's really a very a pathway which has been elegantly elucidated by this gentleman, and that's why he won the Nobel Prize, Aziz Sanchar from the University of North Carolina. He did a lot of work on the biochemistry of this pathway and elucidated a lot of these steps.
Excuse me.
Yes.
Does this happen perfectly more often than not, or?
It's always perfect.
It can be.
Oh, you mean, is it always?
Does it always go to the end and everything?
Yes.
It's obviously
well,
no,
you know.
but how often does it not?
Okay, that's a good question, because I say it's for. I mean, I can't say that every single time. Maybe as far as we know, once in a normal cell, once the process begins, it will end. Now, obviously, shit happens, right? I mean, but there's nothing that really interferes with it. Yeah, but there are people who are deficient in this, who have there's a genetic disease, I'll mention that a little bit later, where it doesn't happen at all, or the efficiency is much lower. We'll talk about that, but it pretty much, once it's initiated, it's very fast. It happens in a few milliseconds, and it will completely remove the lesion. As far as we know, it's virtually 100% error-free.
Is it related to P53 and checkpoint?
Yeah, well, that's some of the work I did many years ago. Our lab showed that P53 regulates nucleotide excision repair. So P53.
Because some cells can escape from this, and that causes cancer.
Yeah, it can escape from
repair, DNA damage, or sometimes they have
they can
mutations.
No, well, you see, if DNA repair isn't as efficient as it should be, and DNA and you get replication across the damage before it can be removed, that causes mutations. So anything that will inhibit DNA repair will, or won't completely bypass it, but if repair is inhibited, that causes cancer. That's the whole idea. So the physiological importance of these pathways, the global pathway, okay, these are just statements, okay, just to make it more clear. So the global repair pathway removes CPDs from anywhere within the nuclear genome. It's relatively slow. You get about 50% lesion removal within about 24 hours post-treatment. But this pathway is nonetheless critical for preventing mutations and thus for protecting against skin cancer, because it removes these very highly mutagenic lesions, helix distorting lesions, whether they be CPDs or induced by other lesions induced by other mutagens. Although it's relatively slowly, it's from the whole genome. So that will, in the case of CPDs, is critical for protection against skin cancer. On the other hand, the transcription-coupled repair pathway, as I mentioned, much more efficiently repairs helix distorting lesions only along the transcribed strand of active genes. And you can get, as we see in cells, up to 80 to 100% removal within 6 hours. So this is very, very rapid pathway. And this preferential repair of the transcribed strand is obviously not very important for preventing mutations in skin cancer, because only about 3% of the genome, I put 5, I mean, it's not 3 or 2 or 3% of the genome is being transcribed at any given time. But rather, this is critical, transcription-coupled repair, for facilitating the recovery of transcription after UV, thereby promoting cellular survival. Because if you don't quickly remove DNA damage from the transcribed strand, there will be no transcriptional response to UV, or no effective transcriptional response. So you have to remove this damage very quickly to allow cell survival after UV treatment. So there's another pathway called translesion synthesis. To put it very simply, translesion synthesis occurs where you have a cyclobutane dimer on the strand, and the other strand is being replicated by a semi-conservative polymerase, and it becomes strongly blocked. Replicative DNA polymerases epsilon, delta, and alpha cannot bypass cyclobutane dimers, just like RNA polymerase II. It's an absolute block. But there is something happens. They only discovered this about maybe 15 to 20 years ago. Something happens which allows a nucleotide to be incorporated at some point, or misincorporated, and then you have extension past the lesion. So there's something that allows DNA replication to recover if the damage is not removed. So translesion synthesis, which allows something, or a polymerase or something, to copy over the lesion and complete replication of the genome, it's not a mechanism of repair, but it's more a mechanism of tolerance. So you don't remove the lesion, the cell just replicates past it, but it remains there, so it increases cell survival, but this is at the expense of advanced mutagenesis. So unlike nucleotide excision repair, which is error-free and rather perfect, this is what we call an error-prone pathway. It deals with DNA lesions in such a way that allows the cells to survive, but it causes errors to be introduced. Now, obviously, a cyclobutane dimer is not always going to base pair with the correct base because it's, whoops, what happened here? Oh, because you have interference with the Watson-Crick base pairing, and you're going to get often misincorporation events here. So it's an error-prone pathway. That's how you get UV-induced mutations.
So all of this happened during transcription?
No, this is DNA replication.
Yeah. Because what about those polymerases? They have proofreading activity.
Yeah,
nuclease
they do, but none of that works. None of that works here. You're talking about pol epsilon, which has proofreading activity, for example, right? But once you have one of these helical distorting replication blocking lesions, the semi-conservative DNA polymerases cannot go past. So you need something to go past, maybe another polymerase. And that's exactly what it is. Mostly in the 90s, they discovered 15, or I think maybe now there's a few more, 15 or 18 distinct DNA polymerases in human cells. And it's these Y family polymerases that are important here in terms of human survival. So you have the replicated DNA polymerases, which are part of the B family, delta, alpha, and epsilon. These replicate undamaged DNA extremely accurately, as you all know. They occasionally make mistakes, but we need to have mutations for evolutionary purposes. But in general, replicated DNA polymerases replicate undamaged DNA extremely accurately. But as I mentioned, they're completely blocked by cyclobutane dimers. They can't get past. So around 1998 or so, they started to discover these Y family polymerases. The main ones are pol kappa, eta, and iota. So it turns out that pol eta, they showed, can replicate undamaged DNA, but extremely inaccurately. It's about 100,000 times. It makes about 100,000 times more mistakes than replicated DNA polymerases. So you don't want pol eta to be replicating, obviously, undamaged DNA. However, pol eta incorporates nucleotides opposite CPDs, so it's able to go past the CPDs very efficiently. It can very efficiently, unlike the replicated polymerase, very efficiently bypass the damage, and surprisingly accurately, so it will often incorporate the correct base. When I say surprisingly, it's really quite amazing because the helix is quite distorted, but pol eta seems to be able to often insert the correct base. So if you have a TT dimer, it will often put in AA opposite and keep going. But sometimes it makes mistakes, and that's how UV-induced mutations are caused. The other Y family polymerases, pol kappa and iota, are also able to efficiently incorporate nucleotides opposite CPDs, but extremely inaccurately. So they make an incredible number of mistakes. So obviously you want pol eta to be on the template. That's what the cell wants. The cell wants to survive. It wants to bypass the cyclobutane dimer, and it doesn't want to make a lot of mistakes. So how does it do this? How does it know, if cells can know anything, which polymerase to use? So the answer is that there's a very elegant mechanism based on ubiquitination. And the way it works, basically, it was discovered by this gentleman, Alan Lehman. He probably should have won a Nobel Prize too, but you can't give everybody a Nobel Prize. So the way this works is here you have pol epsilon on the leading strand. It's happily moving along. It's an undamaged DNA template. It's incorporating nucleotides. You have PCNA, which is the heterotrimeric, the homotrimeric sliding clamp that slides along the DNA, and different replication factors are tethered to it, and it's kind of a platform for DNA replication factors. So pol epsilon is associated with PCNA, and at this point PCNA is not not ubiquitinated. It's in an unubiquitinated state. Pol eta, which is in the vicinity, is actually monoubiquitinated by this E3 ligase PRH2 on 4 lysine residues that actually block a PCNA interacting domain on Pol eta. So Pol eta has this PCNA interacting domain, and it's blocked by ubiquitination. When you have UV, what happens is that PCNA becomes ubiquitinated by this E2, E3 ligase complex, RAD18, RAD6. It's ubiquitinated on lysine 164. And the Pol eta monoubiquitination is downregulated. The mechanism isn't exactly clear. It's probably deubiquitinated by deubiquitinating enzyme, thus exposing here a PCNA interacting domain, which is normally blocked. Once this happens, there's what we call the polymerase switch. So monoubiquitinated PCNA can then interact with the PCNA interacting domain of Pol eta, and Pol eta is Pol epsilon is replaced by Pol eta as the DNA template in front of the lesion. It's really quite an amazing mechanism, which was very eloquently demonstrated. Although some people say it doesn't happen. There's always people who, it's amazing. Anyways, but there's some beautiful papers that show this mechanism whereby Pol eta is switched onto the template preferentially over iota and kappa and replaces Pol eta. So then you can have lesion bypass and resumption of normal DNA synthesis. So Pol eta is able to easily bypass the cyclobutane dimer, mostly inserting the correct bases. So you have the translesion synthesis occurs. The ubiquitin is then removed from PCNA very quickly by a deubiquitinase activity called a deubiquitinase called USP1. The multi-monoubiquitination of Pol eta is upregulated, so it's ubiquitinated by PRH2. This is activated again, and it blocks the PCNA interacting domain. So then PCNA and Pol eta dissociate. As soon as Pol eta bypasses the lesion, then Pol eta detaches quickly from the template, and Pol eta reattaches, and Pol eta mediated DNA synthesis is quickly restored, which is what you want. So Pol eta is able to switch onto the template, bypass, and then Pol epsilon quickly switches back, which is because you don't want Pol eta to stay there too long. Okay, so we'll leave helix distorting, replication blocking adducts for a moment. And we'll talk about modified non-helical distorting DNA bases and single-strand breaks, which are repaired by another DNA repair pathway. So oxidized bases come from endogenous sources, reactive oxygen species from oxidative respiration, because we all know oxygen is poison. When oxygen is metabolized, you get tons of reactive oxygen species. And then pharmaceutical companies expect you to take vitamins that scavenge reactive oxygen species, and they tell you that that's good for you. That's complete bullshit. But anyways, reactive oxygen species do come from oxidative respiration and also inflammatory processes, and they are able to damage DNA quite readily. Well, ROS interact with everything, and also from exogenous sources, ionizing radiation, ultraviolet light, and some chemotherapeutic drugs can also induce reactive oxygen species. And the typical type of DNA adduct, this is 8-hydroxyguanine, which you can see is simply the addition of a hydroxyl group on the 8th position of guanine. So it's not a very big change. It's not helical distorting. Another consequence of endogenous ROS I mentioned earlier are single-strand breaks with unligatable termini. They could be ligatable as well, but for example, you can get a single-strand break that has a 3' phosphate instead of a 3' OH, so you have 2 phosphate groups that can't be easily ligated. So this can represent a problem for the cell. There's also alkylated DNA bases, which can come from some endogenous sources. It's not clear. The metabolism of S-adenosine methionine is thought to induce alkylated bases. But there's different types of alkylating agents. We call them monofunctional alkylating agents. It's not so important, MMS, MNMG, which are monoalkylating agents that are used to study the alkylation process. And there's also chemotherapeutic drugs like temozolomide and procarbazine, and mainly different alkylation products, but the major one is 0,6-methylguanine, which, as you can see, is also like the 8-hydroxyguanine, is the addition of a methyl group on the 0,6 of guanine. So this also is not a very big, bulky modification, and it's not helical distorting. And then you can have so-called spontaneous base damage. So spontaneous base damage arises from the instability of covalent bonds. So cytosine can very rapidly deaminate to uracil, which obviously will have mutagenic consequences because cytosine pairs with G, and uracil, which doesn't belong in DNA, pairs with A, so you can get mutations. I didn't mention here that alkylated bases, there's other alkylated bases. 3-methyladenine is one, which is important. I'll mention why in a minute. And another kind of spontaneous base damage would be simply the hydrolysis of the N-glycosidic bond that holds a base onto the DNA backbone, which results in the elimination of the base to form an A-basic site. So a pyrimidine or a purine base, which is here, will simply be cleaved off through hydrolysis of this N-glycosidic bond, and you get a space in the DNA where there's no base. Now these lesions, they're not helical distorting. Obviously uracil doesn't belong in DNA, but it's not going to distort the helix. But these lesions are highly mutagenic because they don't distort the helix. DNA polymerase can easily bypass them, but they're highly mutagenic. So 8-hydroxyguanine, normally G will pair, with, of course, with C, but 8-hydroxyguanine 50% of the time will pair with A instead of C. 0,6-methylguanine will pair with P instead of C, very frequently. So these lesions are highly mutagenic. However, these other products, okay, and also you get a lot of these. This is another important point. These are produced in the cell. There's a lot of these lesions. Single-strand breaks with unligatable termini, you get 100,000 per cell per day. These A-basic sites, you get 10,000 per cell per day. And these products on the right, which are indicated with arrows, very primitively, also happen to block replication. So this is very important for this talk later on. Single-strand breaks with unligatable termini, if they're not rejoined in a timely manner, and DNA polymerase happens to arrive here, and it's polymerizing, it can't get past this, obviously. And this has to be repaired. So single-strand breaks cause a lot of replication stress, and if they're not repaired and the polymerase stalls for too long, then you can get double-strand breaks forming, which is going to kill the cell. 3-methyladenine also blocks DNA replication very strongly, and of course needs to be removed. So in order to relieve replication stress, you need to be able to repair these lesions. And the pathway, which was basically pioneered by Thomas Lindahl at the you have a double-strand break, and it just ligates them directly, and it's repaired. And the way this works is you have a double-strand break, which is detected and tethered, meaning tethered means that the ends are directed towards each other by this Ku heterodimer. You may have heard of some of these proteins already. The Ku heterodimer, Ku70, Ku80, bind to the ends and bring these ends close together. Then you have the recruitment of an important protein, DNA PK, which is related to ATM and ATR, actually. and it undergoes an autophosphorylation. So DNA-PK phosphorylates itself, and this stimulates the recruitment of enzymes that will process the ends in preparation. It's a kind of DNA resection, which entails the removal of non-ligatable groups to facilitate the ligation. Then you need some DNA repair synthesis, and then once the DNA repair synthesis is done to fill in any small gaps or any small overhangs, you can have direct ligation of the double-strand break. Now this process almost always results in the deletion of a few bases. It's almost never perfect. It's an error-prone process. And as a matter of fact, many of you who might be immunologists know that this mutagenic non-homologous end joining is essential for diversification during DDJ recombination. So non-homologous end joining is a way of quickly repairing double-strand breaks, but it almost always makes mistakes. I won't say always. Certainly almost always. It's a highly error-prone process. So it joins together the ends, and there's always deletion of a few bases. Of the cell cycle phases. So obviously it's particularly important for repair of double-strand breaks in G1 when homologous recombination cannot occur. So it is a major pathway to repair double-strand breaks at any point in the cell cycle. And really what it is, as the name implies, non-homologous end joining, there's no need for homology, and it entails the direct ligation of broken DNA ends. So you have a double-strand break, and it just ligates them directly and it's repaired. And the way this works is you have a double-strand break, which is detected and tethered. Tethered means the ends are directed towards each other by this Cu-heterodimer. You may have heard of some of these proteins already. Cu-heterodimer, Cu70, Cu80, bind to the ends and bring these ends close together. Then you have the recruitment of important protein DNA-PK, which is related to ATM and APR, actually. And it undergoes an autophosphorylation. So DNA-PK phosphorylates itself. And this stimulates the recruitment of enzymes that will process the ends in preparation. It's a kind of DNA resection, which entails the removal of non-ligatable groups to facilitate the ligation. Then you need some DNA repair synthesis, and then once the DNA repair synthesis is done to fill in any small gaps or any small overhangs, you can have direct ligation of the double-strand break. Now this process almost always results in the deletion of a few bases. It's almost never perfect. It's an error-prone process. And as a matter of fact, many of you who might be immunologists know that this mutagenic non-homologous end joining is essential for diversification during DDJ recombination. So non-homologous end joining is a way of quickly repairing double-strand breaks, but it almost always makes mistakes. I won't say always. Certainly almost always. It's a highly error-prone process. So it joins together the end, and there's always deletion of a few bases. So I'm not going to talk about this actually. Okay, I'll talk about it very quickly. Interstrand crossing, because it's not important for the rest of the talk. So it kind of interrupts the talk. So maybe I'll just not talk about it. Okay, you're not responsible for interstrand crossing repair. But it is a very interesting pathway. And if you want to know more about it, I suggest you read this one of these reviews. This is the most recent review. It's really a very interesting pathway, and it's a very important. It repairs covalent links between strands. So if you have two DNA strands, it's not like a CPD, which is a covalent link between bases in the same strand. Interstrand cross-link repairs a covalent link between bases in opposite strands. So the strand can't even separate during replication, so it's extremely lethal, and it needs to be repaired, but it's quite complicated, and I will skip over it. To make life easier, because it's a lousy name.
I want to get home. It really is lousy outside. Okay, so you're not responsible for interstrand prospect repair, and it kind of breaks up the top, but I wanted to talk about it because it's very interesting. Anyways, okay, so we just talked about double-strand break repair, non-homologous end joining, and homologous recombination. So, now that you know about that, I want to make a statement that, okay, DNA repair is a critical determinant in cancer development. So if you go back to the first slide, I showed you how DNA damage drives multi-stage carcinogenesis, but really part of the proof of that it's critical in cancer development, is the existence of DNA repair deficient genetic syndromes. Now, all of these DNA repair pathways, some of which I didn't talk about today, but aberrations in these pathways are often compatible with life, but they cause exquisite cancer predisposition. So there's a disease called xeroderma pigmentosum, which is could be a mutation in any one of these genes. If you go back to the slides, you'll see that I didn't emphasize too much, but these are NER genes that are involved in GGNER or PCNER or both. And people who are mutated in these genes have about up to 10,000-fold increased predisposition to skin cancer. 10,000-fold. So you may have a child who's born with it, who goes into the sun, like often they're normal, except for the, some have neurodegeneration. If any of these XP patients are perfectly normal until they go into the sun for 2 minutes and they can get a severe sunburn, they'll turn as red as this sweater here. Like literally, I wish I show some pictures in some of the other courses, but they're incredibly sensitive to sunlight and to skin cancer. Individuals who have mutations, also who are defective in translesion synthesis, who are unable to perform this relatively error-free bypass with POL-eta, are also exquisitely sensitive to skin cancer. In fact, POL-eta deficient patients are called xeroderma pigmentosum variant. They're not deficient in nucleotide excision repair. They're deficient in translesion synthesis, but because they're unable to deal with UV-induced CPDs, they also have a very similar disease phenotype, a striking predisposition to skin cancer. There's a disease associated with defective non-homologous end joining called ligase 4 syndrome. So these patients are unable to ligate the broken DNA ends. They get a very high frequency of lymphoid tumors, and also because non-homologous end joining is critical for diversification of T cells, they're extremely immunodeficient, and they also have other developmental defects. Then these two pathways, I can talk about interstrand cross-link repair and Lynch syndrome, which are defective in mismatch repair, but patients who have deficiency in these pathways, again, exquisite sensitivity to various tumors. People deficient in basic excision repair can have colon cancer, and of course BRCA1 and BRCA2, and other homologous recombination enzymes that regulate homologous recombination cause this exquisite sensitivity to breast and ovarian cancer, as I talked about. So sporadic cancers, and so these genetic syndromes are kind of proof of the importance of DNA repair in cancer prevention. But sporadic cancers in the general population are also initiated by inactivation of somatic cells of DNA repair pathway genes, or of oncogenes and tumor suppressor genes that lie upstream of DNA repair pathways. So you can get a cell which has like a p53 mutation, for example. P53 mutated cells can't perform nucleotide excision repair, as well as p53 proficient cells, and their p53 deficient cells are very susceptible to transformation by ultraviolet causing skin cancer. Okay, so DNA repair is a critical determinant in cancer development. We all know that, I emphasize that, but that's not the main point I'm trying to make. DNA repair is a critical determinant in cancer treatment. Now, as I mentioned very early, many widely used chemotherapeutic drugs, they can be extremely effective effective in preferentially killing rapidly dividing tumor cells, because many of these chemotherapeutic drugs block replication. They lead to collapsed replication forks, double-strand breaks, they induce apoptosis. But this won't happen in quiescent normal cells, which aren't replicating their DNA. So many of these chemotherapeutic drugs that are DNA damaging agents will preferentially target rapidly dividing tumor cells, and they do this by blocking DNA replication and causing highly lethal double-strand breaks. But as I mentioned earlier, obviously there's major side effects that involve normal cells, especially ones that undergo cell division, such as blood cells, cells in the gut. So if you interfere, if you cause problems with DNA replication of blood cells, you can get obviously immunosuppression and anemia. If you interfere with cells in the gut, that causes severe nausea that's associated with many chemotherapeutic drugs. And also, of course, as I mentioned, secondary tumors, because these drugs are generally genotoxic and mutagenic. So there's a need for approaches. Although these chemotherapeutic drugs are often very effective and they can cure people, as I said, they can also kill people. Just the treatment, as you know, the treatment itself can kill people or make people extremely sick. So there's a need for approaches that more selectively target tumor cells versus normal cells to reduce this treatment-associated toxicity and possibly to enhance the efficacy of the drugs. So the question is, how can DNA repair defects, which characterize many types of tumor cells, be exploited to preferentially target such cells for destruction? So many cancer cells have DNA repair defects, and normal cells don't have them. Normal cells that are not transformed in the patient. So the answer to that is synthetic lethality. How many people have heard of this? Okay, I'm shocked that only one. Like, this is the trendiest thing today in cancer treatment. So you're learning something trendy today. You also, you look like a trendy guy, so yeah. I shouldn't say. No, but synthetic lethality really is one of the major avenues that are being explored for cancer therapy. So this basically, as I said, involves exploiting the weaknesses of cancer cells for therapeutic benefit. It may not be DNA repair. It could be some other pathway, metabolic deficiencies that are exploited. But today we're going to talk about the use of synthetic lethality to enhance therapeutic efficacy of cancer. DNA repair to enhance therapeutic efficacy against cancer. So it's very simple. You have a normal cell, and when this normal cell divides, you can get normal daughter cells. Okay, the normal cell will have two DNA damage response pathways. This is hypothetical, A and B, and these pathways are important for repairing a certain kind of DNA damage. So pathway A and pathway B are critical for repairing whatever, CPDs or double-strand breaks or whatever. So when these cells divide, you can get lots of normal daughter cells that have both pathways, but occasionally you'll get one that undergoes a mutation, either a spontaneous mutation or maybe it's exposed to a mutagen in the environment that will cause the loss of pathway B. So this daughter cell is left with only pathway A, and if it undergoes some kind of genomically unstable, and there might be a few other mutations that occur, it's already initiated, it can turn into a cancer cell. So you have a cancer cell that has lost pathway A and pathway B. But you have the daughter cell, okay, if you treat these cells with a pathway A inhibitor, the cancer cell doesn't have pathway B anymore, and you knock out pathway A, but the normal cell, you knock out pathway A, but it still has pathway B. So it will survive, whereas the cancer cell will die. Is that clear? I mean, it's very simple. It's so simple I didn't explain it properly. So you get it? Okay. It's really that simple.
So I have a question.
Yeah.
Is the mutation or the
if that answers your question.
It depends on them.
It depends on the gene, the pathway, and whether it's dominant or recessive.
Yeah, but the cell is not like.
Or haploinsufficient.
Yeah, the endocyte is not rely on the normal.
Well, if pathway B is haploinsufficient,
And we restore back.
yeah.
Like we have better.
Right.
Rely on the.
Well, it can if the heterozygous situation doesn't confer a defect.
Mhm.
But I'm talking about in this hypothetical situation, somehow you're losing pathway B. Somehow it's lost. So it's not necessarily a mutation. It could be something upstream. It could be a P53 mutation, for example, which weakens nucleotide excision. So somehow it loses pathway B, and it only has pathway A, whereas the normal daughter cell has both pathways, and then if you use an inhibitor, if this grows into a cancer, and then you use an inhibitor of pathway A, this cell will survive because it still has pathway B, which is able to deal with it.
Because sometimes you might have one mutation that does not cause cancer, but with replication and with age, it becomes more, because some people, some doctors say, like, all of us, we have mass cancer, but it depends like, how it can escape and re-amplify and be exposed as chemotherapy.
Yes, of course. Yeah. He's making life complicated, but he's right. Okay, so I'm trying to keep this as simple as possible, but you're right. I understand exactly what you're saying. So, actually, I'll talk about this Nature paper, and first I'll talk about the observation. Okay, so we're coming back to PARP and BRCA1 or BRCA2 deficient cells. So there was a Nature paper published in 2005. Actually, one was on BRCA2. The results I'm going to show you are on BRCA2, but there was an accompanying paper that was published right next to it, where they looked at BRCA1 deficient cells. So this caused the pharmaceutical companies to go berserk. Okay, we're going to see why in a second. So essentially, what you have here, these are killing curves, standard killing curves. On the x-axis, you have on this axis you have increasing concentration of PARP inhibitors. So remember, PARP, poly-ADP ribose polymerase, that's the enzyme that regulates single-strand break repair, religation of unligatable single-strand breaks. So PARP, you treat cells with PARP inhibitors. So these cells here are wild-type cells or BRCA2 deficient cells that are complemented with BRCA2. So either wild-type or BRCA2 complemented with BRCA2. You can see that in response to PARP inhibitors, there's really no difference in killing. So PARP, at very high doses of PARP, you're going to get some killing. Even here you get some killing. With this particular inhibitor, there's not much killing except at this dose where suddenly you get some toxicity in normal cells or BRCA2 complemented cells. But look at these are BRCA2 deficient cells. Look at this killing here, especially here. This is unbelievable. So you have a minute dose and the cells are completely wiped out. So BRCA2 deficient cells and BRCA1 in the other paper are incredibly sensitive to PARP inhibitors. Incredibly sensitive. And then in another experiment in this paper, there were more experiments, but the key ones is they showed that this particular PARP inhibitor specifically kills BRCA2 deficient tumors in a mouse xenograft model. So here you have days after implant. Here you have, this is essentially a measure of the size of the tumor. Again, you have BRCA2 complemented cells and BRCA2 phenotype or BRCA2 BRCA2 deficient, or BRCA2 complemented cells with inhibitor, you get this, the tumor grows in the same manner. However, a BRCA2 deficient cell, BC8, treated with a PARP inhibitor is completely destroyed. So this was the first indication that PARP inhibition and mutant BRCA1 or BRCA2 exhibit synthetic lethality in a preclinical model. So when you treat mice with a PARP inhibitor and they have a BRCA1 mutation, that completely eradicates xenografted tumors. And this suggests a highly targeted and minimally toxic anti-cancer treatment. And then subsequent studies showed that cells exhibiting what they call the mutant BRCA-like phenotype, which are essentially cells deficient in homologous recombination. So cells deficient in proteins like NBS1, ATM, CHEK1, RAD51, RAD54, PALB2. You remember, these are the proteins I told you have to remember that are involved in homologous recombination. These cells are also sensitive to PARP inhibitors, but especially BRCA1 deficient cells are exquisitely sensitive to PARP inhibitors in these kinds of experiments. So how does this, what was the rationale for these studies? So PARP is a DNA damage sensor, as I mentioned earlier, that binds with high affinity to single-strand breaks, after which it rapidly transfers poly-ADP ribose polymers to the cell. So PARP is poly-ADP ribose polymerase, and that's what it does. It transfers these polymers of poly-ADP ribose to itself and to other proteins. So it auto-poly-ADP ribosylates itself. This facilitates the recruitment and poly-ADP ribosylation of DNA repair proteins, including those basic scission repair proteins directly involved in the repair of unligatable, replication-blocking single-strand breaks. So these poly-ADP ribose chains can then be removed by an enzyme called PARG, poly-ADP ribose glycohydrolase, and single-strand break repair can proceed. So this is how single-strand break repair occurs, PARP-mediated single-strand break repair works. So also, I guess, as rationale for the previous study, PARP-deficient cells, according to this, are defective in the repair of unligatable replication-blocking single-strand breaks, and as a result will exhibit increased replication fork collapse leading to double-strand break formation and cell death. So if you have single-strand breaks that are being produced constantly in the cell, we call them spontaneous because they're not caused by an outside mutagen, they're caused by reactive oxygen species that are produced in the cell. And if you take away PARP, the cell suddenly becomes defective in the repair of these replication blocking single-strand breaks, and that will they'll accumulate, and you'll get replication fork collapse and double-strand break formation. But PARP deficiency is not deleterious to normal cells. So PARP knockout mice don't accumulate mutations in the cancer and are fully viable. But in the presence of BRCA1 and BRCA2, which, as I mentioned, are required for efficient, error-free repair of replication-associated double-strand breaks, there's a problem. Okay? So, model for the selective killing, this is also very simple, for selective killing of BRCA1 or BRCA2 deficient tumors through pharmacological inhibition of PARP alone. So you just treat, all you need is PARP. You don't need other DNA damaging agents. So you administer a PARP inhibitor to cells or to a patient. This will result in the accumulation of single-strand DNA breaks, many of which are unligatable. During DNA replication, the overwhelming number of single-strand DNA breaks, they can't be repaired. They're going to collapse into cause collapse replication forks, causing double-strand DNA breaks. In a normal cell, which has BRCA1 and BRCA2, these double-strand breaks can be repaired very efficiently by homologous recombination, which allows survival. In the case of BRCA1 deficient cells, or cells that are deficient in other homologous recombination proteins, there's no repair. So you still have accumulation of these single-strand breaks and double-strand break formation, but there's no opportunity to repair them. And because double-strand breaks are so lethal, the cells die at a very high rate. So this is synthetic lethality. A PARP inhibitor will not affect normal cells that have homologous recombination, but a PARP inhibitor will destroy cells that are deficient in homologous recombination because they have no way to deal with double-strand breaks. So a question which arose, the other pathway, non-homologous end joining, so it's error-prone. As I mentioned, it's thought to play a major role in DNA repair, especially when homologous recombination is gone. So is it possible that non-homologous end joining might protect homologous recombination deficient cells against the cytotoxic effects of PARP inhibitors? That you would logically think that they could rely on non-homologous end joining, but that's absolutely not the case, because it was shown just about after that non-homologous end joining actually drives poly-ADP ribose PARP inhibitor lethality and homologous recombination deficient cells. So when they treated BRCA1 deficient cells with PARP inhibitors, that induced the phosphorylation of DNA PK substrates, stimulating error-prone non-homologous end joining, and this enhanced the frequency of mutations of chromosomal rearrangements. So aberrant activation of error-prone non-homologous end joining could be one of the causes of genomic instability leading to cytotoxicity in homologous recombination deficient cells treated with PARP inhibitors, and this was quite unexpected, but quite an important finding. Now, as I said, the pharmaceutical companies went berserk with this. They saw these data, preclinical mouse models and with cells, and you simply administer PARP in a BRCA1 deficient situation, and you get complete destruction of cancer cells, whereas normal cells aren't affected. Of course, they got very excited. They started to spend hundreds of millions of dollars on clinical trials with PARP inhibitors, but there's a problem, because the main sponsor PARP treatment, using a drug called iniperin, which was Sanofi Aventis. These were the major clinical trials, and there's a lot more. These are just a few of the clinical trials. There's really a lot more. It was discontinued because after using it on many thousands of patients for 2 or 3 years, they found that it wasn't this particular PARP inhibitor. Now, what does that mean? I don't know what that means. That's ridiculous, right? I'm just trying to tell you this because I don't like the pharmaceutical industry. So, anyway.
Because some of them they do that.
Well,
They get some money and they refund it.
Well, no, it's just that they saw these incredible results, and they should do clinical trials, but they rushed it. Okay, and the people who discovered the PARP inhibitors, the people who published those Nature papers, they rushed to form companies to make PARP inhibitors so they could sell them to the pharmaceutical company, and everybody was so excited. Actually, there was another PARP inhibitor, olaparib, they used it for a while. It didn't really work very well. Some of these clinical trials, PARP inhibitors did have a positive effect. So you may have had patients whose life was extended, maybe for a few months, which is important. I'm not saying it's not important to extend somebody's life for a few months. It's very important. But we're talking here about spending hundreds of millions, if not billions of dollars, and they use this olaparib. They're all ended in olaparib, and it didn't work very well, so they discontinued it for 2 years. And then people started to get really, the public especially, said, how can you discontinue this? It works in some people. So now they recontinued. They're doing clinical trials with it again. It's not really clear. Here's synthetic lethality does have a lot of potential in principle. If you can treat cells with a drug that will specifically knock out a DNA repair pathway, or any pathway that's required for tumor cell survival, but normal cells haven't, that's the best way to treat cancer, one of the best ways to treat cancer selectively. But they're really struggling with this part, and there's a lot of money being spent on it. CIHR funds many grants that look at PARP, and I'm not so sure that it's going to end up not being a flop. But anyways, it is a very interesting example of synthetic lethality. So people are now looking. So whether or not PARP ends up working, people are now it's the rage to. One way to do this. There's many ways now to do it. People are using CRISPR-Cas9 methods. This is a little bit more of this was used, let's say, in the around 4 to 5 years ago. People still use this approach. They take isogenic cell lines that are wild type or mutated for critical cancer-associated genes. For a critical cancer-associated gene, it could be P53, for example. And they fluorescently tag these cell lines differentially with different fluorescent tags. So you have the wild type cells and the mutant cells, like the mutant P53. They mix them together in 200,000 1-6 wells, they have these big robots. And then they use RNAi or small molecule compounds to screen for lethality. And if there's no effect, obviously both of the cell types will grow. If it's lethal to only one, either the mutant or the wild type, only one of the cells is very easy to screen for by fluorescence methods. And if it's lethal to both, all of the cells will die. So really what you want are treatments that are selectively lethal to the mutant cells. So here, they've identified quite a few potential synthetic lethal compounds or pathways, whether it's small molecule compounds or RNAi. Obviously, you're getting different information when you use either of those. But so there's now this big drive to spend more hundreds of billions of dollars screening all of these promising compounds that give synthetic lethality. So I think that's it. Yeah, so the conclusions are obvious. DNA repair status is a critical determinant in cancer development. So I think I gave you a lot of evidence why that's true. But it's also a critical determinant in cancer treatment, and especially with respect to synthetic lethality. What I was telling you about PARP, I'm a little bit cynical about it, but that doesn't mean that this synthetic lethal approach is not a very promising way to treat cancer. So, end of slideshow, click